This is the last chapter that's included in the A&P 1 curriculum, and it's over special senses, and we're kind of getting close to the end, so we don't have a lot of time to put into this chapter. So I'm just going to go over the basic highlights with you to give you enough information to carry forward and also give you what you need for the comprehensive final. So there are four major senses. We have smell, taste, hearing, sight, and then we also have equilibrium. Those receptors are also included in the same area where the hearing receptors are. But we're going to start with chemical senses that are called taste and smell. Taste is also called gustation, and olfaction is the other word for smell. Now, these receptors that respond to the stimuli that are chemical in origin, and so they're called chemoreceptors. And the definition of a chemoreceptor is a receptor that responds to chemicals that are dissolved in an aqueous solution. We'll start with taste. Taste is picked up by organs embedded in the tongue. Most of them are in the tongue, and these are called taste buds. These are the receptor organs. There are about 4,000 taste buds on the tongue, and they're in structures called papillae. You have fungiform papillae on the top of the tongue. On the side walls of the tongue, you have what are called foliate, and then in the back part of the tongue, you have these taste buds that are called circumvallate papillae. Now, these are the largest of the taste buds. They're quite large, and they have a V shape it's behind the anterior two-thirds of the tongue. It kind of separates between the anterior two-thirds and the posterior one-third of the tongue, so they're way in the back. There's a few taste buds on the soft palate, cheeks, pharynx, and epiglottis, but most of them are found on the tongue. There's a look at the tongue showing you fungiform papillae at the top, these linear shaped foliate papillae at the sides, and there's your circumvallate back here. Lingual tonsil is behind that V shape of your circumvallate papillae. Here's the palatine tonsil and the epiglottis. So the structure of a taste bud, actually you have 50 to 100 flask-shaped epithelial cells of two types. One are actually receptor cells. These are gustatory epithelial cells. Those are your taste cells. So it's unusual that you have epithelial cells actually being sensory receptors. We always think of nerve tissue being involved with receiving sensation. This is an exception to the rule. And these epithelial cells are replaced every 7 to 10 days. Now, there are microvilli or gustatory hairs that are the actual receptors that are attached to these cells. And there are three types of gustatory cells. One releases serotonin, others lack synaptic vesicles, but one releases ATP as a neurotransmitter. And these basal epithelial cells are the dynamic cells. These are like the stem cells that divide every 7 to 10 days and give rise to the actual taste cells. So here is your taste bud. There are the gustatory hairs. This is the epithelium of the tongue. And then this is the connective tissue underlying it. And here are your taste fibers of the cranial nerve, because cranial nerve 7, 9, and 10, remember, are associated with some aspect of the sense of taste. There's your gustatory epithelial cells. There's the taste pore. And there's your basal epithelial cells that give rise to the new functioning taste cells or gustatory cells. And there's an enlarged view of a taste bud magnified 210 times. So taste is really affected by smell. 80% of taste is smell. So when you have a cold and your nose is plugged up, you really have a hard time tasting things. There are some other aspects of taste. There are thermoreceptors that respond to temperature. And because when food is warm and hot, a lot of these, the steam that comes off the food makes those chemicals airborne, then you smell those vapors, and that's what enhances the flavor of the food. So warm food tends to be more palatable than cold food. There's also mechanoreceptors that respond to texture. So when you have grease that is melted and warm, it's more palatable than if you were to eat a hamburger where the grease is solidified and is slimy. So that has a lot to do with the appeal of certain food items. And nociceptors are pain receptors that also influence taste. So some things like real spicy food and hot sauce and things like that, to some people, initiates a sense of pain, burning. You know, some of the peppers that are out there can be pretty distasteful. Some people like them, but they can initiate nociceptors and pain receptors in some people. So temperature and texture do enhance or detract from taste, like I said. Now we're going to go to olfaction. So this is a look at your olfactory nerve, and we have branches of the olfactory nerve that go down through the openings in the cribriform plate. Remember, that was in the ethmoid bone. And so your smell receptors are in the upper part of the nasal cavity. So in order to really smell something, you have to get these odorant molecules up into the upper part of the nasal cavity. So that's why you sniff to smell something, get those odorant molecules and chemicals up closer to those nerve endings. So if you look at this a little bit closer, you'll see that this is the olfactory epithelium, and your olfactory cells are actually bipolar neurons that are embedded in these supporting cells, and then there's a mucus lining. So here's your olfactory cilia, there's the dendrite of your bipolar cells, this is your olfactory sensory neuron, and then those pathways go up through the cribriform plate, and they form little round structures that are called glomeruli, and those attach to mitral cells that end up making the olfactory tract. So the bipolar cells to the mitral cells. So that's the pathway of the olfaction pathway, rather. Now we go to sound. Now we said that taste and smell, those are chemoreceptors, okay? So you're responding to chemicals that are dissolved in an aqueous solution. Now when it comes to sound, you're talking about electromagnetic waves, okay? Part of the electromagnetic spectrum. So when you have a tuning fork and you tap a tuning fork, you're making these little prongs go back and forth. And when the prong goes this way, it compresses the air. And then when it pulls back, it separates the air. So you have these areas of compression where the molecules are pushed together, and where they're apart, those are areas of rarefaction. Okay, there's the word rarefaction right there. So high pressure where the molecules are pushed together, low pressure where they're apart. So that can correspond to waves. And where you have the high pressure, that is a high wave right there. That's where the wave is at its peak. And then where the pressure is low, that's where the trough is of the wave. So you see how the wavelengths correspond to those different areas of high pressure versus low pressure. Okay, so the top is the crest, the trough is the bottom. Now, depending upon the frequency of the wave, the frequency is perceived as pitch. And frequency means the number of waves that pass a certain point in a given period of time. So if you compare the blue to the red, you'll see that the blue is a low pitch because you have only 3 going through this 0.03 seconds. Now the high pitch is the red because you have 1, 2, 3, 4, 5, 6 waves going through in the same period of time. So the higher the pitch, the faster the vibration, and the lower the pitch, you don't get as many waves per given period of time. Now the loudness of the wave is also the same as amplitude. So the higher the wave, the louder the sound. And if you think about ocean waves, you have those nice gentle waves that don't really carry a lot of energy behind them, but if you have something like a tsunami, a huge wave, it's carrying a tremendous amount of energy and is a much louder, carries a lot more energy behind it. So higher in amplitude, a much larger wave. So you can see that here. Okay, so the ear has three major areas. The first one is the outer ear, which is for hearing only. The middle ear is called the tympanic cavity, that's hearing. And the inner ear has hearing and equilibrium structures. And the receptors for hearing and balance respond to separate stimuli, and they are activated independently. So here's a look at the outer ear. The outer ear itself is called the auricle or the pinna. The round part is the helix. The bottom fleshy part is called the lobule. And this is the external ear. Then it goes into something called the external acoustic meatus, sometimes called the external auditory meatus. And so your external ear actually ends, if you look at this bracket up here, it ends here where the tympanic membrane or eardrum begins. The middle ear includes the tympanic membrane and this air-filled cavity here that contains three little bones. And the three little bones in the middle ear are called the malleus, the incus, and the stapes. And then you go into what's called the internal ear or inner ear, which is a labyrinth. It's like a little maze of twisted channels. And this is the inner ear structures right here. Now, at the bottom of the middle ear, which I said again is an open air-filled chamber, is this pharyngotympanic or auditory tube that's usually in a closed position, but enters into the lateral walls of the nasal cavity, so it is something that can help to equalize air pressure, which we'll talk about in a minute. Those are just your main structures. So here you see the inner ear structures. There's the cochlea, that's for hearing. So hearing receptors are in this part. There's the cochlear nerve. There's the vestibular nerve. And remember, number 8, cranial nerve number 8 is called the vestibular cochlear nerve because it has branches that are attached to equilibrium receptors, which are here in the semicircular canals and the vestibule. So equilibrium receptors are in those two areas, and their impulses are picked up and carried through this vestibular nerve. And then the cochlear nerve picks up receptions that are interpreted as sounds, okay, sounds that you take back to the brain and ID those. Again, this is your middle ear, tympanic membrane, the three little bones, the malleus, the incus, and the stapes. There's your spelling, and there's your pharyngotympanic tube. So we're enlarging here the membranous labyrinth of the internal ear. So these little channels that look like a maze almost, they are enclosed in bone. And this is in the petrous region of the temporal bone. And the actual structure of or size of the cochlea is about half the size of a split pea. So it's very, very small. And it's coiled. And these structures here, your semicircular ducts, we're going to talk about these in a little bit more detail later, but these are for dynamic equilibrium when you're spinning in space. And then you have what are called the utricle and the saccule. Those two are involved in static equilibrium, so that you can tell where you are when your eye's telling you you're standing still but you're moving, like being in an elevator or on a boat or something like that. So now this is looking at the cochlea and cutting a little section out. So you'll see the cochlear nerve here, and you'll notice that there are these little chambers inside of the cochlea. Now I'm going to make these a little bit larger so we can get more detail. So inside the cochlea, you'll see that there are open areas. And the top one is called the scala vestibuli. Now perilymph is a fluid that is very much like cerebral spinal fluid. It's very, very thin fluid. Endolymph is fluid that you see here that's in this middle chamber that they put the word out here because this is small, so they couldn't fit it in here. But this is called the scala media or cochlear duct. This contains a lot of potassium because potassium is going to be the electrolyte, or kind of like a neurotransmitter. It's going to work as a neurotransmitter that helps to pass along the impulses. So potassium is very important in here. And this scala tympani is very much like the perilymph, is the very, very thin water-like fluid that you find is the same as what's in the scala vestibuli. Okay, so those are your three major chambers. Now, this is called the organ of Corti. This is where your hearing receptors are actually located. So here are your hair cells that pick up the receptions. Those are the cells, and here are your supporting cells. And this is the basilar membrane that your hearing receptors are actually resting on. And then this is a branch of the cochlear nerve that picks up those receptions from the hearing cells. Now this tectorial membrane is what the cells are attached to. So what's going to happen is you have sound waves that come in, and these waves will cause a vibration, and that's going to cause the tympanic membrane to vibrate, and that's going to cause these little bones to move. And when these little bones, which are collectively called auditory ossicles, but individually it's the malleus, the incus, and the stapes, but collectively the auditory ossicles. Now they're going to pass that signal along to the stapes, and the stapes is bound to this structure that's called, it's a membrane that's called the oval window. This is an air-filled chamber. Remember, I talked about this earlier. Over here, it's fluid. And when you pass along a signal from air to fluid, it amplifies the signal. Besides the fact that you're going from a larger surface area to a smaller surface area, so you amplify the sound. So the pressure waves created by the stapes pushing on the oval window move the fluid in the scala vestibuli. Now, if you know anything about fluids, they're not compressible, okay? So you have to have a pressure release valve somewhere, and that's going to be right here at the round window. So as this is vibrating and pushing the sound waves through the fluid, this is going to vibrate on the other end to release that pressure that occurs here in the fluid when it's introduced. So sounds with frequency below hearing travel all the way around through the other end, which is called the helicotrema. Now remember, this thing is coiled up. They've just stretched it out for so they can show you exactly what's going on here, to make it easier to see. And they don't excite the hair cells, okay, because they're too low. Sounds in the hearing range go through the cochlear duct, vibrating the basilar membrane and deflecting hairs on the inner hair cells. So what happens is, when you move that basilar membrane, it bends the hairs that are attached to that tectorial membrane. You can go back and look at the picture again. And that's going to, the bending of the hairs is what passes along the signal. And it's through the bending of the hairs that opens up potassium channels that causes the depolarization of the membrane and passes along the signal. So here it is again, okay, where you have the outer ear, the pinna. Okay, here's your vibration coming through. You go through the auditory canal to the tympanic membrane. Remember, I said this is a larger surface area to a smaller area, so you get an amplification of the signal, also going from air to liquid. And you go through the little bones, amplification in the middle ear, oval window, and then you go through the fluids of the cochlear duct, and then wherever that signal is picked up, whichever area of the organ of Corti is stimulated, that's where you get that pitch of sound that is passed on to the temporal lobe of the brain where sounds are interpreted. So the pathway of sounds, here's your basilar membrane. So high frequency sounds actually are picked up in this area. And if you look down here, you'll see that in the beginning of the cochlea, the fibers are short and stiff, very tight fibers, and that's your high end. That's 20,000 hertz. Down at the other end, these are long, floppy fibers, about, going down to 20 hertz, and so your low sound is here. So if you think of a guitar, your high-pitched strings are the very tight, thin strings. The ones that give you the lower sounds are the looser, floppier, fatter strings, and so it's kind of the same idea. And a harp, this kind of looks like a harp, same idea. Tight, short strings, high-pitched, long, looser strings, low-pitched. So the beginning of the cochlea is where you get your high pitches picked up. And then when you get across to the middle, medium frequency, and then down at the other end, low frequency. So because all of your sound has to come through the beginning, most hearing loss occurs with your high frequency pitches. That's where you get the condition called presbycusis, which is old people's loss of hearing, and it usually happens in the high frequencies first. So here's the pathway mapped out for you. Sound waves pass through the auditory canal to the tympanic membrane. Vibration of the tympanic membrane causes ossicles to move, sets up the amplification of signal, and vibration of the oval window. Sound waves pass through the fluids in the cochlear canals, causes movement in the basilar membrane. Bending of hairs in different regions of cochlea represent the various pitches of sound. High pitch close to the oval window, low pitch close to the apex. Pressure caused by vibrations released by the round window, impulses generated in the cochlea, and then cochlear nerve, and then from there you go to the cochlear nuclei in the medulla, inferior colliculus. Remember we talked about the reflex center, auditory reflex center, which is in the midbrain, and auditory cortex in the temporal lobe, which provides conscious awareness of sound. Whew, that's a lot, right? Okay. So, processing. Pitch perceived by impulses from specific hair cells in different positions along the basilar membrane. We already talked about that. High pitch close to the beginning, low pitch on the other end. Loudness is detected by bending more hairs. So the more action potentials you pass through, louder deflections of those, or more bending of the hairs, allows more of a depolarization and louder signal. More hairs are affected. Localization of sound depends on relative intensity and relative timing of sound waves reaching both ears. Your ears can tell that the sound's coming from the right because it reaches the right ear before it hits the left ear. Even though it seems like it's instantaneous, there is a timing difference that your brain can pick up. Now, there is types of deafness. Conduction is where you have some interference with the sound actually getting to the cochlea. So you could have a problem like impacted earwax. Sometimes that happens as people age. Some people are prone to having problems with producing a lot of earwax. Perforation of the eardrum. You can't get the eardrum to vibrate if it's inflamed or if it's damaged. And then otosclerosis is a hardening of the ossicles, where they don't actually move. Sensory neural deafness, tinnitus is ringing of the ears. That usually indicates some type of a damage to the hair cells, or maybe part of the cochlear nerve somewhere along that pathway, but it could be an indication that there is some hearing loss. Some medications, like if you take a lot of aspirin, can cause tinnitus as well. Meniere's syndrome is a labyrinth disorder and a buildup of fluids, and a disease that sometimes affects, usually it's unilateral, but it can cause vertigo and a lot of annoying sounds in the ear, and it can be mild to severe depending upon the person's individual display of that disease. Okay, so let's look at equilibrium. These are structures that are associated with static equilibrium. These are what we call the macula, and the macula has two structures called the utricle and the saccule, and you'll see them here. Now the saccule is kind of straight up and down, and the utricle is kind of horizontal. And these structures have hairs embedded in them with supporting cells. And then they have this sort of a jelly-like membrane that has these otoliths, which are calcium carbonate salts embedded in this jelly-like matrix. And so what they do is that they respond or they move depending upon which way the head's moving. So if the head is moving to the right or left, those little stones will bump into the hairs and will cause you to perceive that things around you are moving. And there's also, before I move on to the eye, the semicircular canals respond to rotational movements of the head. Okay, so the eye and vision. 70% of the body's sensory receptors are in the eye. That's amazing to me. And visual processing really makes up about half of the cerebral cortex. So the eye is really the windows to the brain, and the brain really has a huge amount of its functioning devoted to visual input. Now, most of the eye is protected by a cushion of fat and bones, which is the orbit, so it's a very delicate structure and it's very well protected. Parts of the eye, there's three tunics. The fibrous tunic is the outer part. The sclera is what we call the white of the eye. The cornea is the transparent anterior part, and that lets light into the eye. The vascular tunic, and you can see down here, I've drawn a little picture. It's not perfect, okay, but there's the sclera, there's the cornea in the front. Underneath that in red, that is your vascular tunic, also called the uvea. Now, the choroid layer is dark brown. It absorbs light. It also has the blood supply for the tunics. And the ciliary body is in the front. That's smooth muscle. So here's the choroid layer. The ciliary body is right there. And then anterior to that is the iris. And the iris is the color portion of the eye. And the pupil is the central opening. Then you have the very innermost sensory tunic, which is the retina. That's the neural layer. This is where your photoreceptors are found. They're called rods and cones. And they are attached to bipolar cells, which are receptor cells that transmit the impulses from the rods and cones, and then they go back to ganglion cells whose axons form the optic nerve. So there in green is the retina, there's the optic nerve, there's the optic disc called the blind spot, there's your choroid body underneath, okay? Green, retina, red, choroid body, and sclera in white. So those are all the main layers. This picture gives you a lot of information. It shows you the sclera, the choroid, the retina, the anterior chamber. This is the cornea here. This is the anterior chamber in here. This is the posterior segment that's filled with vitreous humor. That's a jelly-like fluid. The anterior is more like a water-based fluid. This scleral venous sinus is the drain for aqueous humor. And there's your pupil, there's the iris, and showing you suspensory ligaments. There's the lens of the eye, there's the ciliary body, so all the basic parts of the eye. Focusing for close vision. Light from close objects less than 6 meters diverges as it approaches the eye. So in order to see things close up, the eye has to make active adjustments. There's three things that happen. Accommodation of the lens. Okay, so the lens is going to round up. Constriction of the pupils, so the pupil gets smaller. And the eyeballs converge. It's like when you bring your finger close to your eyes look like they're crossed because they're convergent. They're getting closer to the middle. So those are the three things that have to happen to accommodate for close vision. This is looking at focusing on things close up, and this is what's going on with the eye. So it's also a parasympathetic response to contract the ciliary muscle. So by contracting the ciliary muscle, you are loosening, allowing the lens to bulge. Okay? So contracting the muscle, this is almost like a sphincter, because remember it's 360 degrees. So you're bringing this closer together, and that's what makes the lens bulge up. And remember, when you're going through convex lenses, that's convex, this is convex. The image actually is inverted. So focusing on distant, this is the sympathetic activation. The lens flattens. Okay, so relaxes the ciliary muscle. It opens up, it flattens the lens, and now you can see things far away. So close up, rounding up of the lens, flattening out is for seeing things far away. So emmetropic is the term given for normal vision, where the eye is perfect in its orientation, enabling it to take in light rays, converge them exactly onto the focal plane of the retina, so it meets right there, and you're able to see the image clearly. Hyperopic eye means that you're farsighted and the eyeball is too short. And the focal point now, it doesn't mean your eyes are flat. We're talking about just slight. You can't really tell visibly. But the focal point goes behind the retina. So the corrective lens is a convex lens, which is going to converge the rays just a little bit more to pull them in, so that now your focal point is exactly where it should be and pulls the focal point forward. The myopic eye, where you're nearsighted, the eyeball is just a little bit too long, and so the focal point is ahead of where it should be. So the corrective lens is a concave lens, and with a concave lens, you diverge the rays out a little bit more so that they focus back behind where they normally would, so that you can see the object. This is a look at the retina. And so you see these are your photoreceptors. There are your cones and there are your rods. And the pathway of signal comes from this direction. And the pathway of light comes here. So you go from your photoreceptors to your bipolar cells to your ganglion cells, and the axons of the ganglion cells will eventually exit out the back of the eye as the optic nerve. So light adaptation. Sensitivity of the retina decreases sharply as you adjust to more light. Retinal neurons switch from rods to cones, so you're bleaching the rods and you're activating the cones, and usually takes about 60 seconds. Going from light to dark, cones stop functioning in low intensity light. Rod pigments that were originally bleached out by bright light and inhibited, they start to become activated, and rhodopsin accumulates in the rod and sensitivity of the retina increases. This usually takes longer than it does for light adaptation because you have to get the rhodopsin put back together and activated. So that's the difference. So your rods are a lot more sensitive than your cones, and they so that's the difference between light and dark adaptation. Now, nyctalopia is the term given for night blindness, and a common cause is vitamin A deficiency. So photoreception, rods are very, very sensitive to light. They absorb all wavelengths of visible light, but their inputs are perceived only in fuzzy gray tones or fuzzier images. If you want to see color, you need to activate cones. They have very low sensitivity, and they contain pigments that allow for color perception, a sharper image. These are your wavelengths that respond to your different colors, blue and green and red. So this is showing you the colors of the electromagnetic spectrum broken down and photoreceptor sensitivity. There's your actual electromagnetic spectrum going from radio waves all the way to gamma rays, and showing you where the visible light spectrum is. So activating certain cones will give you the range of colors that you see. And depth perception is when both eyes view the same image from slightly different angles. Depth perception, three-dimensional vision, results from cortical fusion of slightly different images. And you can see this with magic eye pictures that were popular several years ago, where the picture looks like a collage, and then if you let your eyes relax and fuse the images together, you get a three-dimensional picture. And it requires input from both eyes. And that finishes up a very, very brief look at the special senses. And there's a lot more to this, and if you want to spend time with any or all of the senses, they're very, very fascinating. So that ends this chapter.